Hello, everyone. Whoever is joining us today, we have a good paper presentation. Tanu, stage is yours. No, I think we can start. Yeah, all good. Tanu, why did you choose this paper again?
Include a lot about the interaction between microbiota and host and the immunological responses from it. So I was just going through some papers and I thought this paper looked really interesting. So here is it today.
Okay.
Through
Yeah,
some paper.
I like this paper. I will not pretend to understand all of it, but
I'll try my best today. Let's see. This is a bit away from my comfort zone, so it's not a virus paper. But because we have a lot many virologists in our journal club these days, I was like, let's try something new.
yeah, we have a lot of T-Reg people and a lot of virologists. Maria is the only one who's not a virologist or not a T-Reg person.
Well, actually, in my department, a lot of people work on T-Rex. Maybe not right now, but they used to in the past. So maybe I will get to know some more about T-Rex soon.
Well, that's what we need. Another person about who works for T-Rex.
No, need diversity.
Yeah, we need diversity. We need to get some people who work with agar plates. Anyway, Tanu.
Okay. The article I'm presenting today is Microbiota Targeted Maternal Antibodies Protect Neonates from Enteric Infection. This was published this year in Nature by the first author is Wen Zhang, if I'm pronouncing it correctly. And let's dive into it to see what this paper says. This paper uses a pathogenic organism for its studies, and that's enterotoxic E. coli or Escherichia coli, and I'll be referring it as ETEC for the rest of my presentation. Uh, so acute infectious diarrhea is one of the major causes of deaths, especially in children below the age of 5. And annually, they can cause about 1.5 million deaths. And that's that number of deaths only by bacterial diarrhea. There's also viral diarrhea, which is caused by rotavirus and norovirus, which is also a field of my study. And that also causes a great deal of deaths in children below the age of 5, especially in the developing countries. So this is a region of concern. For bacterial diarrhea, ETEC along with Vibrio cholerae and Shigella are the main culprits causing symptom of diarrhea. And ETEC generally causes diarrhea in infants, which is below the age of 2 years.
Do they get it from polluted water?
Polluted water can be one of the reasons, but also food, sanitation, condition, hygiene, everything matters. In this study, they also use neonates, mouse neonates, as their model organism. And why neonates? Uh, because they are highly susceptible to microbial infection and they infect these neonates or their mothers with the ETEC and do the further studies. And why they are highly susceptible to microbial infections is because they have a immature immune system, which is less adaptive to immune effectors. And the second thing is they lack diverse commensal microbiota. So it's over the years you are living that your microbiota gets more and more diverse. But when you're just born, there is a narrow range of your commensal microbiota.
Yeah, I have one thing to add here about the neonates and having an immature immune system. That the vaccines that are used for neonates, they are sometimes different from the vaccines used to immunize adult people.
Mhm.
And you would often see that protein vaccines, or I mean polysaccharide vaccines, they would be conjugated with a protein, because the polysaccharide response in neonates, it's very poor. So if you just give a polysaccharide as a vaccine, they won't get immunity. So you would sometimes conjugate it with these proteins to make them more immunogenic, but in adults you don't need that because the adult immune system is much more capable.
Mhm. The passive immunity for this neonates can be obtained through maternal antigen-specific IgG antibodies or through vaccination. And till now, in the general world, vaccination has been considered as a way of immunizing them. But there are studies which are considering the maternal route to be a better option, and this study is one of them. But what are maternal and natural antibodies? In this article and as well as in my presentation, they will be referred to as mNabs. So, maternal natural antibodies are antibodies which are acquired without known exposure through any pathogen or through immunization. So, they are what are already naturally present in the mother or in any human. But they are called maternal because they can be postnatal, they can be transferred to the babies prenatally or postnatally. So prenatally, they can be transferred through placenta or postnatally through breast milk. And how the mNabs diversity is induced is unknown, and how mNab work postnatally, that is via milk, is still unknown. So this is when I say milk, I mean breast milk or breastfeeding. It has been seen that breastfeeding can reduce overall rates of diarrhea and mortality in the developing countries, but the mechanism behind it are not yet researched or not very clear. Diving into the experiments now, there are two things to keep in mind before we go into the protocol of this article. There's lots of genetics, but the two things to keep in mind for every experiment is mu-empty knockout mice, which is this one, lacks mature B cells. So you can see there's no antibody in the drawn on the mouse over here. So mu-empty knockout mice won't have any mature B cells, but mu-empty like normal mice will have mature B cells. And the
Tanu, one
second
second.
thing, yeah,
Just since we are here and we are trying to be as educational as possible, anybody wants to shed some light why what is mu empty and what's the knockout doing to the B cells? Anybody wants to take this one?
you can go ahead.
Okay, I'll just go ahead. Okay, so mu empty. This is the heavy chain of the antibody, and this is the M chain, and this is the first one that's expressed on any B cells. B cells cannot survive if they are not able to express a functional B cell receptor on their surfaces. So without the M chain, every time this B cell will go through gene rearrangement, since there is no M chain, it can never express the first B cell receptor, and it will never mature. These B cells, they are born, but since they don't mature, they just go through apoptosis. So it's a cycle of getting born, like taking birth and dying right there. So that's why these mice will never have a mature B cell. I don't know how the heterozygous work though. Maybe half of those have it, or I don't know if it's codominant or dominant
I
expression.
think they have just enough number of mature B cells to protect them from any infection.
Okay, so there are fewer B cells in these heterozygotes.
I'm not very sure whether it's fewer. I think it's optimal or enough.
Okay.
Okay. Okay, as I was saying, the first thing to remember is that the mu-empty knockout mice won't have mature B cells. And the second thing to remember is for the first 3 weeks of birth, the presence of IgG and IgA in neonates will totally depend on whether their mother has mature B cells or not. In this case, this is a mother, and it's been bred with a knockout father. So the F1 genotype will have both knockout mice as well as the normal mice. But for the first 3 weeks, all the mice, including the knockout mice, will have B cells. As you can see, the drawing of antibody on top of them will have B cell present in them. Sorry, will have IgG and IgA present in them because they are being breastfed by the mother, which has mature B cells. But after 3 weeks, when they are adult, now the adult knockout mice doesn't have endogenous antibodies, but the normal mice does. Any doubt in this figure? Okay. Now, if the mother is the knockout one and the dad is the normal one, the babies, whether they are normal or knockout, won't have any antibodies in them for the first 3 weeks of their birth. But when they're adult, the normal mice themselves can produce B cells and antibodies now, but the knockout, as usual, won't have any antibodies in them. So it's easy. Mu-empty knockout won't have any antibodies. Mu-empty knockout babies won't have any antibodies. But if it's a normal mice, the babies, whether it's a mu-empty positive or knockout, will always have the antibodies in them.
So are the antibodies coming from the mother not persistent for a long time? Like after 3 weeks, are they suggesting that they're gone?
I think they're suggesting it's only through breastfeeding. That's why after 3 weeks, the mice doesn't breastfeed anymore. It depends on the food, and that's why there's no more postnatally transferred antibodies in them.
Right, but even after 3 weeks, they would still kind of persist. I mean, I don't know what the half-life of antibodies are, like passive antibodies. So
I think 3 weeks, that's a lot of time for antibodies. Like that's more, that's enough time because normally in our body we have the B cells that are constitutively making.
yeah.
So as the diet changes, the mice stops breastfeeding and goes to food diet, and hence the antibody in the mice also stops, like transferring from the mother to the kid. Pups for the mouse. In the very first figure, okay, let me tell you what's the experiment they did. This is the breeding that's happening. And in the first study they did was to confirm if the immunization with mNabs will work against pathogenic infection for these pups. So they won't be letting them grow. They are doing all their tests in these pups within like 1 week to 10 days, while all their the presence of the antibodies in them is totally dependent on whether their mother has antibodies or not. In the first study, they are just checking the immunization with mNabs, and they're using 6 to 7 days old mice, and those mice were subjected to human clinical isolate of the ETEC strain 6. And as I said already, the ETEC stands for enterotoxic Escherichia coli. This ETEC6 can colonize the small intestine of neonates and cause acute and lethal diarrheal disease within just 20 hours of oral inoculation. In figure A, at sublethal dose, which is like 10 to the power 6 or 7 of ETEC, the mNab plus pups, that is the pups which had antibodies in them, was more resistant to ETEC infection than mNab minus pups. And they basically had, so the ETEC colonization is measured in these experiments via looking at the bacterial burden in the small intestine. They can see that there is a 33-fold reduction in the ETEC colonization in the intestine, which is very significant. So they are saying that yes, the immunization via maternal antibodies is working.
I wonder if these antibodies, are they just blocking or neutralizing these pathogens, or are they actually causing phagocytosis and adaptive response?
That is a good question, and that's not in the study, but that's a good thing for their future studies, I guess.
Because if it's just neutralizing, then I think it's easier for the baby. It's less, there is no fever response, there is nothing. You just have neutralized antibodies, and now nobody wants to look at them. But yeah, still this result is something. How did they put these antibodies again? mNab positive, did they give these antibodies as an injection?
It's via breastfeeding.
Okay.
So the mothers which are mu-MT positive will have
Okay.
mNab plus pups. The mothers which are mu-MT knockout will have mNab minus pups. Now at higher dose, all the mNab plus pups were still resistant, whereas 83% of the mNab minus pups died or were almost dead. 20-hour, these experiments are done within 20 hours because in 20 hours, the mouse is supposed to die if they have this ETEC colonization in their intestine. But 20 hours is not at all enough for antigen-driven endogenous IgG or IgA production. So IgG or IgA, anything present in this pups at this time will only be maternally derived via breastfeeding. Now they are testing the IgG in serum and in small intestine. And as expected, the mNab plus pups, which is the pups from normal mouse, do have IgG present in their serum as well as in the small intestine. But on the other hand, the mNab minus pups from the knockout mice do not have any IgG present either in their serum or in their small intestine.
Has anyone here ever done IgG in small intestine? I've never done these experiments, but I would think it's very difficult because you would have to homogenize all the tissue. It seems like a lot of technical challenge.
But it's important for these experiments because the ETEC is colonizing in the small intestine. So you have to see whether in the final target place the IgG is there or not.
Yeah. Also, they are working with pups, and if they have to take the intestine, that means the euthanasia is required. And I would hate to be euthanizing pups because you can't even put them in a CO2 chamber. You have to behead them.
let's not go into the ethical concerns.
Yeah, it's cruel.
Don't make
That's
me
all I'm
feel
saying.
bad for choosing this paper.
Oh, the science is good, but that's the price we pay for.
That's required. I'll tell
Yeah.
you why in the discussion.
Also, they're doing these experiments within 20 hours, which means they're basically doing it in a day. I don't know how they're doing
They're
all
not
of
going
this.
home. They're not going
They're
home.
not going home.
They're staying there in the
Or
animal
they're
house.
they're working in shifts. That's possible.
That's possible, yeah.
They're like, hey pups, as long as you are alive, I'm staying here. No
Yeah.
worries. Now they check if the presence of IgG is anyway related to the commensal bacteria, which is already present in these pups. So the commensal bacteria from mNab plus pups, which can be seen via in the red, were found to be correlated with IgA as well as IgG. So this bacteria did have IgA or IgG attached or associated with their surface. This shows that the maternal IgA and IgG, which can be associated with commensal bacteria from gut microbiota, are undergoing the vertical transmission from mother to child via breast milk. Now the figure F, it shows that only IgG was found to be associated with the ETEC cells itself. So from the gut microbiota, they separately took out the ETEC cells because those were the pathogen they inoculated and those can colonize the small intestine. So on the ETEC cells, now only IgG was present, but no IgA was present. Moreover, it was confirmed that the differential protection against the ETEC was not due to gut microbiota compositional change, as both the mNab plus and mNab minus neonates had similar composition of gut microbiota. So whatever is happening because of the antibodies and not because of the influence of gut microbiota on the immune system.
I'll make an honest confession right now that until this paper, I did not know that IgGs were transferred from breast milk. I thought it was only IgAs.
That's true. Like that's the common notion that because IgA is a secretory antibody, not only, but the majority of the immunoglobulins in milk will be IgA, which also is true. I think the majority is IgA, but not every time. It's not the IgA every time, which is helping to stop the pathogenic infection. Like in this case, I think it's only the IgG and not the IgA, which is stopping the ETC
Yeah,
infection.
because the IgA can cross the epithelium with the polymeric Ig receptor, but for the IgG, I I don't know how much that IgG Fc receptor is expressed there, so it would be difficult, right?
Yep.
But yeah, let's see
Let's see.
this paper.
Yeah. By these experiments, they have confirmed that the protection is conferred to breastfeeding pups only. Breastfeeding pups having a normal mother only, although the mother was not previously exposed to the pathogens. The next thing they did was a cross-fostering experiment. In this case, they refer fostering to as whose milk the pups are drinking. So when they mean cross-fostering, they mean that the pups having normal mother are drinking knockout mother's milk, but the pups having the knockout mother are drinking the normal mother's milk. So here are the mNab plus pups from the normal mother, and here are the mNab minus pups from the knockout mother. And as soon as they were born, they were taken away from their mother and they were cross-fostered. The mNab plus pups now have the knockout mother, and thus they do not have any antibodies in them right now because their breast milk don't have any antibodies in them. So this mouse will be calling as plus to minus in this presentation, where in the other hand, the mNab minus pups were cross-fostered by the normal mother, and now they have antibodies in them. So we'll be calling them minus 2 plus. So did not have previously, but now they have. And this were had previously, but now they don't have because their mother don't have in their breast milk.
So they don't get antibodies from the placenta?
They did get the antibodies from placenta, but that's basically gone as soon as they're born. And
I guess
now
that's
all
the
the
control.
antibodies away.
And it's not that. I wonder how they did this too. Like, I don't know if they have like a mini mouse breast pump or something, but you know, it's not the easiest thing in the world to get mom mice to foster other non-pups, because as we know, working with mice, like the females are much more aggressive during pregnancy and post-pregnancy than the males are. Like in fact, sometimes you actually have to separate the pups from their moms because they can be so aggressive sometimes, and a lot of times they can also sense whether if there's a defect in the pups, and they will cannibalize them too. So it's like, I think also we even had a litter or a particular strand of mice where we couldn't even touch the pups because even our scents to rub off on the pups will be translated to the mom that that's some form of danger. So to take mice and essentially give it a surrogate to breastfeeding, it's like I really wonder how difficult it was to actually do these experiments, because that's a miracle.
Yeah, I think maybe you'd have to anesthetize them, because this is something we discussed in a past podcast, that B6 mice, they will accept any new pups within the first 3 days of birth, but if you introduce any pup after the first 3 days, they will not take care of that. But yeah, the scent is also a thing. I don't know how they take care of the scent. Maybe they're wearing a lot of gloves and no perfume. The experimenters.
It's a real sensitive job though, honestly. Like even to wean our mice, and if we have mice that has 2 litters in one cage, and you have a younger litter of pups and maybe a bit older litter of pups, and you want to separate the cages out, even that's a tricky task, because these moms really, scent wise, they really pick up on stuff, and they really pick up on just the foreignness from their own being. So I don't know, I commend this group if you can figure it out. But I know some strain of mice are more docile, but still, it's tricky.
Wait, were these bulb C? Let me check. Because bulb C, those mice, they actually are more accepting when it comes to fostering other pups, because yeah, again, in that podcast we discussed, they mentioned that bulb C mice actually are open to taking new pups within the first 7 days of giving birth, compared to 3 days.
I'd even like to see that in person. I'd even like to see that.
Yeah, all these pregnancy related experiments, they're so sophisticated and probably require a lot.
But if that's true that they can take it within the 7 days, they have done it right in the day of their birth. So I think that's why the mothers were fostering other pups.
Okay, it's B6 mice. These are not the
Okay.
cute bulb C that let you do.
We are wrong then. Don't really know how it happened, but it happened. The mothers did foster those pups, and now they either have the antibodies or do not have the antibodies. But now this new cross-fostered group of pups were orally inoculated with high dosage of ETEC6. Now let's see what happened. The -2 plus pups, that is, which this -2 plus is they didn't have antibodies, now they have antibodies, and the -2 plus will be did have antibodies, now don't have antibodies. As we can see in this figure, the -2 plus pups had high IgG titers in the serum as well as in the small intestine, as well as high IgA in the small intestine. The bacterial amount in the small intestine of -2 plus pups were much lower, which is 30-fold lower as compared to +2 minus pups. That means the bacterial burden again tells us about how much the ETEC was colonized in the small intestine. And this says that it was either not colonized or very little colonization happened. Also, in the extended figures, an experiment was done where the mNab minus mouse were divided and half of it were given it to the knockout mother and half of them were given to the normal mother. And in that case, 90% of pups receiving the normal mother milk survived after 20 hours as compared to just 20% survival in pups receiving the knockout mother's milk.
Wait, did they not do statistics on GHI, or there is just nothing significant? Because that
What
I, it looks like,
Which GHI are you talking about?
yeah.
I mean, what exactly are you
I
talking
mean, and
about?
there's no, I don't see any
Like
stars
the
there.
figures G, H, and I.
Yeah, and
Oh,
I'm pretty sure some of these, there should be some significance, some of these groups.
Yeah, definitely.
But any case, like if you look at I, it seems like the cross fostering of mu-MT negative to a mother who can make antibodies, it gave this mouse more IgA
more
than,
IgA, right?
yeah, than
Then
normal
actually,
mouse. It's strange. It's very strange.
that's strange. Yeah. I'm pretty sure this will be significant because as you can see, there's like almost no error bars in them. All the mouse had the same result, which is so rare, but
Mhm.
they haven't done any statistical analysis on them. I don't know why.
Although they are using standard error of mean, and that thing seems to, it tends to be tighter than standard deviation.
Okay.
But still, I would expect at least one of these to be, yeah, maybe they didn't just think it was important, which is weird. Why why do you think it's not important to do statistics on these?
Yeah, at least the ETEC burden was statistically significant. By this cross-fostering experiments, they now say that milk derived from the mNabs blocks the ETEC colonization and does protect against the ETEC infection. Basically, it's not who gives birth to who, it's who receives the milk from whom, which is protecting them against pathogenic infection.
This has been a very repeating theme over the papers I have read, that the foster mother has a huge role to play in the, at least in the, when it comes to immune system of the neonate. Because in the paper we discussed, the antibodies from the milk, they were guiding how many Tregs are there in the colon of the in the mouse, in the newborn mouse, and the birth mother had nothing to do with it. It seems like the whole milk process has a huge role to play, and it makes you doubt if, sometimes when mothers are not able to feed their babies and they get the formula, if that is a good idea or not.
That is really interesting. And it also gives out a point that the placental transfer of immunity is not that important as compared to what happens after birth, whether it's milk or the environment or anything which is responsible for inducing any immune response within the neonates are much more vital as compared to what happened before the birth.
Yeah, it could be.
And I also add, just because I am a mother and I talk to a lot of other like physician scientists and other scientists who have children and they breastfeed, what do you guys think when you put in the context of women who decide to breastfeed their children past, even 12 months of age? Sometimes you have moms who elect to breastfeed 2, 3, 4, 5, sometimes years down the line as well, and since you kind of mentioned that this mechanism is proceeding past just this first 7 days of birth, it's like, what do you guys think about that?
Like for mice, it's 3 weeks only when the breastfeeding is actually working. I don't really know like if there is any research for till when human breast milk has all the beneficial quantities.
I feel we would
Right.
need an
Uh,
animal model that mimics the humans in this case, because I don't think we can
Right.
force mice to over breastfeed, right?
uh
I'm not
Oh,
just not sure.
maybe I need to dig more deeper into just what that means. Like, has there ever been any studies that have breastfed mice, or not even breastfed? Sometimes you can just do adoptive, transfers to them essentially past the 3 weeks after weaning. But I'd like to see that. I think that, just this study alone in the first few figures is showing that the antibodies effects from breast milk, really does numbers.
it does. It reminds me
Well,
of the,
when
yeah. You go
well,
ahead.
when do human babies get, like a mature microbiota? Because that will kind of drive whether their endogenous antibodies will be sufficient.
So I guess it depends on what you mean by mature, because soon as baby, well, actually, okay, so it was thought that, baby in womb is microbe free, right? But now
But
there
that's
are,
not true. Yeah.
right. Now there are more recent evidence that there are some forms of microbes even in the placenta, right? So when you think about afterwards, when baby's born, they're exposed to the environment immediately, even in the vaginal canal. So it's like, what is mature micro? What is a mature population? Because even if we wash ourselves and we clean it like to the T, within the first hour, our own like microbial signature will replenish itself in the first hour. So it's like, and that's just surface skin. So I don't know, these microbes are pretty persistent.
Yeah. To add on to that, even the studies that have been done in relation to my lab, it's like even the infection via a virus can change so much in the microbiota. It can alter the microbiota after every infection. And that's what I want to say, that I don't know what a mature microbiota will be, or even if the microbiota is constant throughout the lifetime of a person, because it depends on so many things, like basically everything, every infection, every diet is affecting your gut microbiota, and it changes based on how you are taking care of yourself.
I guess maturity is more just about diversity, right? A lot of these studies, they look at how diverse a person's microbiota is, and then they make inferences in terms of diseases and things like that. So I guess, newborns, they don't have a very diverse microbiota. They might have, quite a number of different species, but I just don't know, like, at what point it becomes more and more diverse. And is there like a threshold when that happens?
Yeah, that will be a good thing to look at. I didn't find that in that study, and I don't have any idea, but that's a good question. Yep. I think that will be the time point when the milk or anything won't be that effective, but I don't know. In the next set of experiments, they use FcRn, which is neonatal Fc receptor expressed on intestinal epithelial cells. This FcRn is the receptor which can transport IgG across the placenta to the fetus. So these are responsible for placental IgG transfer. Or in adult mice, FcRn can transport the IgG from the lamina propria, where all the immune cells in the gut are present, to the lumen of the gut, where the food or any nutrition is present, or any microbes in that case are present in the lumen too. So it transfers from the lamina propria to the lumen.
But wait, when you're having breast milk, isn't it the opposite way? So,
That's what they are doing in the study, yes.
oh, okay.
You revealed the secret.
Oh, sorry. I'm so sorry.
Spoiler.
Spoiler alert. Okay. In this figure A, they again did a lot of genetics. That is, they have the parent mice, which both of them are mu-MT knockout. That means they do not have any mature B cells or antibodies in them. But one of them is FcRn positive, and both of them are normal mouse in FcRn. So both of them have FcRn in them. But the F1 generation now will either have FcRn or will be FcRn knockout. What they do of these pups is all these pups are being fostered by mu-MT plus, which is a normal mother. And now they want to see if in absence of mu-MT, which is B cells, but in presence or absence of FcRn, the antibodies are being transferred to the pups or not.
Tanu, I wonder if they're normal because I still see the heterozygous.
They are heterozygous. You're talking about the mother, right?
Mm-hmm. Yeah,
Yeah.
I don't just from assumption. I think they're still not wild type because normally individuals should have both the alleles. So I think they are like they have reduced expression just so that if you have both the alleles, maybe there is so much IgAs that it's hard to either detect or something. Maybe they want to reduce the expression. That's why they're using these heterozygous models. But yeah, I just want to say I don't think they're wild types.
Okay. I'll not be calling them normal, then I'll just call them heterozygous. Normal was a word I was referring to instead of saying mu-MT plus minus. So I'll call them heterozygous mothers. Yeah, which are breastfeeding all these pups, and let's see what's happening after that. In the figure B, the FcRn minus mice, knockout mice, did not have any detectable IgG in the serum, whereas the FcRn plus mice had significant higher IgG in the serum, although both of them were fed by the heterozygous mother having the antibodies in them. In this figure, they say that the pups having FcRn showed that some IgGs were directed towards the maternal microbiota. By these experiments, they have shown that FcRn is also an important aspect of whether the pups will be receiving the antibodies from mother or not. And that might be because FcRn is transporting the IgG from ingested milk present in the gut lumen of the neonates to serum in suckling mice. So serum will be, I think, not in the lumen, in the lamina
That's
propria.
big news to me. I wouldn't think like FcRn, is it a bidirectional transporter or is it separately expressed on the lumen and separately on the lamina propria side? So
To
many
be
questions.
honest, they also did studies to differentiate the functioning of FcRn in neonates versus adults. And in adults, they did not find this bidirectional thing. So in adults, they only transfer from lamina propria to the lumen. But in neonates, it's somewhat the opposite. It's from the lumen to the lamina propria.
In the discussion, they also mentioned that, but that adult thing could be because the adult intestine is more acidic. There is more acids there,
or
and
have more proteosomes.
it's possible that those IgGs just got proteolyzed before they could reach the
That
FcRn.
can
That's
be
possible.
possible too.
Do they make any give any explanation how IgA is being transported, and if it is in the same way?
But that's for PIGR, right? The polymeric Ig receptor, I would assume.
They're not talking much about IgA here because as they have seen previously, only IgG was getting associated with the ETEC, which is the pathogenic bacteria they're working with in the study. So at a certain point, they just stopped working with IgA anymore, and they're just concentrating on IgG for now.
But yeah, PIGR is also known for transfer from the lamina propria to lumen, but could it take place in the opposite way? I don't know.
I think this was one of the first studies which is showing the bidirectional function of FcRn in neonates versus adults, or in both of them. But more studies based on their mechanism will be really interesting. When IgG binds to the surface of binds to the surface of a bacteria, it can initiate immune effector functions such as complement-dependent bacteriolysis or opsonization. And as previously seen, that IgG and not IgA was coated onto ETEC, they speculate that it is the IgG which is protecting against the ETEC bacteria. So in figure E, the pregnancy of 2 knockout mice were synchronized, and on the gestation day 18 and postpartum day 2, I am not sure why that day is particularly important, but on that day, one of the females received IgG injection, like a vaccination kind of thing, and the other female did not receive it, received a PBS injection instead. Now, the pups from these mothers were challenged to low dose of ETEC6, and in the pups, they have seen that the pups from IgG treated mother had 25-fold lower bacterial load and were highly protected from the infection. And in fact, the serum of the IgG titer of pups fostered by this mother was very similar to that of mNab plus pups.
Sorry, where did they get the IgG for injection?
They purified it from SPF mice, which is specific pathogen-free mice. So this mice was colonized with ETEC, and therefore it had IgG against ETEC. So it purified from the specific pathogen-free mice and was given to the knockout mother as vaccination.
Okay. 5 milligrams? That sounds like a lot to me. I
5
don't know if that's
milligrams is a lot. I don't know how many pups. 6 pups, but still it's a lot.
Like, what kind of concentrate? You would need an insanely concentrated amount, because you can't give more than like 100 microliters IP, right? So I don't know how concentrated this must have been.
Yeah, that's what 100 milligram per ml. That's a lot.
Yeah.
Yeah.
Okay.
So these studies are very important in saying that if you vaccinize the mother in place of injecting the pups, still then you are getting protective immunity against the pathogen. So supplementing IgG alone will give you enough and sufficient protection to the fostered pups from ETEC infection. That's an interesting thing, because as soon as a human baby is born, they are subjected to needles, and that they talk about in the discussions too. What if you give the vaccinations to the mother, can the immunity be transferred via breastfeeding to the kid? This was a really interesting aspect of that thought.
Wait, if I look at it the way that they took the serum out of the SPF mice, because SPF are specific pathogen free, but they're not germ free. They still have
Yeah,
other bacteria.
yeah.
So if you're taking out serum, and there is, I think, 5 milligram or like 10 milligram, or that's not too much for serum, because it will have a lot more antibodies. And after that, the way they are purifying it, I don't think they're purifying it for antibodies that are just against
Oh no, it's the
ETEC.
total IgGs,
So
but
total
I'm
IgG,
saying
that's not a
that
lot in
because
that
they
case.
are specific pathogen free, they were colonized with ETEC so that at least some of the IgGs amongst the total will be against this
Okay.
pathogen.
In that case, I don't think it's a lot, because there's very few antigen specific IgGs in this total. Very
Hmm.
few. I think 1 out of 1,000 even.
Mhm.
Mm-hmm.
Now they see if any commensal bacteria can help induce the mNabs and protect against the ETEC infection. Total ETEC6 directed serum, uh, yeah, total ETEC6 directed serum immunoglobulin and IgG were high in titer in SPF mice mother than in germ-free mice mother. So SPF mothers are in red, germ-free is in black. And as you can see, the total IgG as well as the total immunoglobulin as well as the IgG are higher in concentration for the SPF moms. Um, and also after the SPF mice was serum was absorbed with fecal bacteria completely, they removed of all the protective antibodies. So the protective antibodies were associated with the microbiota, and when uh, the fecal microbiome was given to the serum and then again pelleted it out, now the serum doesn't have any immunoglobulin or IgG present in that. So that's what they mean by absorption. Next, they see that 1 week old mNab plus pups had higher titers of ETEC, specific total immunoglobulin, and IgG, uh, which the serum from mNab minus pups did not. And lastly, they could collect like 2 gram negative enterobacteriaceae. And I again don't have any idea why they chose that very family or phylum to work with, but they could collect 2 gram negative enterobacteriaceae. One was a Pantoya species and the other was an Enterobacter species. And they collected it from the mu-MT plus dams. And now the Pantoya was used to absorb the total immunoglobulin or IgG from SPF mice, and they can see that almost all the total immunoglobulin or IgG were absorbed by just this species itself. Any
What do you mean by
questions?
absorbed?
Okay, that's what I was explaining right here. Is you have the serum of the SPF mice, which does have the total IgG as well as total Ig as well as IgG present in that. Now, if you are giving uh, bacteria which are isolated from the fecals of the mice, you're giving the microbiota, total microbiota or the total bacteria from the feces into the serum, and you're letting it incubate for some time and then pelleting out all the bacteria separately. Now all the uh, IgGs or immunoglobulins which are associated with that fecal bacteria will be attached on their surface and be pelleted out along with them. So now they are just checking the supernatant, and the supernatant was devoid of any total immunoglobulins or IgG. In this case, they are separating only the Pantoia species from the feces and not the whole microbiota. And the Pantoia species by itself can absorb all the immunoglobulins or all the IgGs on its surface and be pelleted out, and the serum will be devoid of any immunoglobulins or IgGs. That's what they're showing
does that
here.
mean these are cross-reacting antibodies between
That
Pantoia and
means
ETEC?
all the antibodies which are being transferred from mother to pups or are there in the mUMT plus mothers are or can be associated with the microbiota of them,
Okay.
which is, I don't know, seems dubious, but that's what's
I mean,
happening.
so this Pantoia species is inducing all these antibodies. Probably that's why these antibodies are coming back to bind to it, right?
That's
Or
what they are suggesting, yes.
okay.
They are suggesting that yes, there are some commensal bacteria which can elicit a cross-reactive antibody against ETEC. But the question now is a single species of commensal bacteria sufficient to protect against ETEC6 infection? For checking that, a vaccination type strategy was applied. So when I say vaccination, that means heat killed Pantoia or heat killed ETEC. Sorry, not heat killed, formalin killed in this case, Pantoia or formalin killed ETEC were given to the germ-free mice dams, which is germ-free mother. Okay, let me rephrase that. Germ-free mothers were immunized with either formalin killed ETEC, which was taken as a positive control because, of course, killed ETEC will be a perfect vaccine for ETEC infection. And they were also non-immunized or unimmunized mice as a negative control. And they are testing formalin killed Pantoia to be to check that whether they are acting as proper vaccines or not. And all the pups were infected after vaccination. So the mother was vaccinated, the mother was germ-free, and the pups were infected with ETEC, and the normal breastfeeding happened. And then this figure show that the survival and the bacterial burden in liver or spleen. So the survival of the Pantoia vaccinated pup, how will I say that? The Pantoia vaccinated mom's pups were almost as good as the ETEC vaccinated mom's pups. That means the vaccination is happening and it's good enough.
So are they in the survival that's an N of 2? Is that what that means?
Oh no, they repeated these experiments twice. In first group, the N was 12 and 7, and the second experiment the N was
Oh, I see. Okay.
so it's these are
It's
averages
yeah.
of the first and second experiment. The
Okay.
dots. I also thought that how do we have an 80% mouse survival?
Yeah.
Doesn't make sense.
Yeah.
So they're basically suggesting there's like some cross reactivity going on here.
They are suggesting that the Pantoia species is inducing the IgGs, which are protective against ETEC infection. So instead of ETEC itself, which is a harmful bacteria or a pathogen, if you just immunize the mother mice with Pantoia species, which is commensal and not harmful, it can give you it can also give immunization to the pups against this infection. And now the bacterial burden is showing how much ETEC was colonized in the liver or spleen. And again, you can see the Pantoya immunized pups were almost similar to that of the positive control, which is ETEC immunized pups in both the liver as well as spleen.
And this Pantoya probably is commensal, as in does not cause
It's the
disease.
commensal.
I'm looking at some papers, and this is a lot of papers are suggesting it as a probiotic.
I really want to know their thought process, why they chose Enterobacteriaceae phylum or Pantoya, and
But all of those are
specific.
gut pathogens, right? That's why.
All of them are commensal. They
Yeah.
they purified everything from the commensal microbiota of this pups or their mother or specific
Did they
pathogen.
use McConkey agar for this for looking at
I have to
Enterobacteriaceae?
read it one more
No,
time.
I think they used McConkey agar, which is such a basic technique, but still gives you gram negative and gut still gives you gut pathogens. So I'm teaching a microbiology course right now, and I'll probably ask my students about this. Why did they use McConkey? Thanks for giving me some idea.
This was my very first time hearing about Pantoya. I have never heard of it before.
Yeah, same.
Okay, so basically Pantoya immunized mice's pups were significantly more protected against ETEC6 than unimmunized mice's pups. That's what these results say. Now, pups born to germ-free mice immunized with Pantoya had cross-reactive IgGs against the ETEC, which can be seen with the red curve, while the pups born to unimmunized mice did not have any antibody present against the ETEC. They say that commensal microbiota can induce cross-reactive protective antibodies against pathogens. That was the last figure, and now we come to the discussions. In this study, we have seen that breastfeeding by dams lacking specific immunity to ETEC can still deliver natural antibodies and hence protect the neonates against pathogenic ETEC infection. The IgG, which was present in the breast milk, reacted strongly with the microbiota of mNab plus pups. And lastly, ingested IgG present in neonatal gut can be taken up into the serum of those neonates by FcRn. FcRn can work bidirectionally across gut epithelium based on age. And I think this will be a very interesting study whenever it comes out, that how it changes over the age or what actually stops it from proper functioning when the mice ages. So lastly, although the authors have not addressed if this milk-mediated pathway is good option for immunization or to be used in therapeutics for human infants, if this technique turns out to be effective and practical, this is a non-invasive approach and it can prove to be so much more advantageous over the conventional needle-based passive immunization strategies. Like there will be so much more anti-vaxxers or just caring mothers in general who don't want the needles to go in their babies will be taking this very approach to immunize their babies instead of the conventional approach. And lastly, I think after all, the world does deserve cute, happy, and healthy babies.
Mhm.
Thank you.
No, after this discussion, I hate this paper less.
Yeah, it was actually very, very interesting.
Mm-hmm.
And you know, when
Straightforward
I
too.
first, oh yeah, very straightforward. When I first looked at the paper, they had 4 figures, and I was like, how did they? I didn't even read it. I just read the abstract, and I saw the 4 figures. I'm like, how did they get into Nature? And then I saw that they have 10 supplemental figures.
Yeah.
Yeah,
10
It's
supplemental
insane.
figures, which have like 10 more within them.
yeah.
Yeah,
No, it
there
was
was
clearly
a
a lot
lot
of
of
work.
supplementary data, as well as the Excel sheets. They have given like literally the whole lab notebook of this project was given to the Nature Journal, and they have published a lot of data, like specific ELISA readings and everything in the
Yeah,
supplement.
I really commend them for all of the work that they've done. This is a really great paper.
Yeah. Thanks, Tanu. Thanks to you, I get to read these papers, else I am always stuck in my own bubble.
I'll stop sharing the screen. Thanks everyone for attending today.
Mm-hmm. Samriti,
I
did you
hope I
say
was clear, like I could help you understand the paper more.
I didn't feel that it was out of your comfort zone.
Mm-hmm.
I did a lot of work yesterday night.
Samriti, did you say you were going to present next?
Yeah, I'll present next.
Okay. Okay, then we'll have some. Are you going to pick some oncolytic viruses?
I don't know yet. I don't
Okay.
know.
Okay, we'll wait for that. Any last thoughts, guys, before we wrap it up?
That was all.
Okay. Thanks everybody for joining us. I'm going to switch the screen. See you all later.